Okay, so welcome everyone to Making It Brain Day 2. It's so nice to see your messages in the chat. I can see some people from Kazakhstan, Poland, Turkey, Scotland. Really nice to see you all here, and thank you again for coming back for the second day. So if you weren't here yesterday, this is what the talks that we had yesterday. And we heard from Tatjana, who talked about biomarkers and early diagnosis of dementia. We had a look around some labs in Cardiff and also at Imperial College London. And then we also heard the clinician's perspective of dementia research. So clinicians, doctors that combine their research and working as a doctor as well. We heard from Sumi yesterday and Nan, who also shared their stories of how they work now in dementia research. And so today we have another jam-packed schedule for you. And today we're going to hear from the following speakers. So we're going to hear from Dr. Alexi Nott, Dr. Blanca, Diaz-Castro, and then flash talks from speakers from UCL and King's College London. And in between, we're going to have a look around more of the UK Dementia Research Institute labs. And so what I really hope is that you from this conference see someone that's perhaps a little bit like you and realize that there's so many different ways of doing dementia research and lots of different journeys into dementia research as well. So if you have any questions for our speakers, please leave them in the Q&A box or the chat, but feel free to use the Q&A. And then I will collate them, and then right at the end of the session, I will put them to the speakers. If you are able to stay to the end, I'll be introducing you to some further resources that we recommend if you're further interested in the journey into dementia research. And these both days have been recorded, so we will be sending you a link to the recordings in your inbox, hopefully tomorrow. So without further ado, it's my real pleasure to introduce Dr. Alexi Nott, who is from the UK Dementia Research Institute at the Imperial Centre. So thank you so much for coming to speak to us today, and leave your questions in the Q&A for Alexi. Thank you.
Hello, hopefully you can see me. So I'm just going to share my screen, share my talk. Okay, so, oh, wait, let me get this full screen. Okay, great. So today I'm going to talk to you about epigenetics, and I'll explain what that is later, immune cells and Alzheimer's disease. But first I'm going to give you a little bit background on my academic journey, so how I got to where I am. So my initial interest in biology started when I was at school. So I grew up in Plymouth. I went to a comprehensive school there. And I was interested in particular what happens inside cells. So I decided to do an integrated master's in biochemistry. So an integrated master's is where you basically do 3 years of a bachelor's plus 1 year of a master's, but all mixed together. And this was great because this course at Bath allowed me to do two 6-month research projects. And I also was able to do both of these abroad. So the first one I did at Duke University. So this is at North Carolina in the US. And here I was working on a research project looking at learning and memory in rodents. And the second project I did at Baylor College of Medicine. This is in Texas, so also in the US. And here I was looking or studying genetics in the context of epilepsy. So I finished my undergrad, and I really liked both of those concepts, so the neuroscience and the genetics, and I decided to kind of combine these for my PhD. So I ended up working on a project looking at neurodevelopment and how epigenetics is important for this to occur properly. I did this under the supervision of Antonella Riccio here in London, so at University College London. And I ended up staying for 5 years with Antonella before doing a postdoctoral research position back in the US. So I keep returning to the US. But this time I was interested more in brain conditions. And still working on epigenetics. So this is work I did at Massachusetts Institute for Technology with Liwei Tsai, looking at different neurodevelopmental conditions and how epigenetics plays a role in that. And then I did one last postdoctoral position. So I did 5 years in Boston at MIT. And then I shifted gears from neurodevelopment to aging and dementia, again in the context of epigenetics. And this is work I did with Chris Glass in San Diego in the University of California. So I switched from the East Coast of the US to the West Coast. And 2 years ago, so following my work with Chris, I actually joined Imperial College London. So that's in West London, based at White City, where I basically started my own group. So this is what my group looks like at the moment. And we're continuing this work looking at dementia and epigenetics, and this is what I'll be talking to you about today. And upon joining the department, I also started this LGBTQ+ allies network. So I identify as an LGBTQ person. So I started this network, and part of the kind of incentive of the network was to promote LGBTQ visibility in the department. And also I'm hoping that we can provide a bridge with the wider LGBTQ STEM community outside of Imperial. Okay, so moving on to the science. So just a quick recap on Alzheimer's disease. So this is what I'm kind of, my research is mainly focused on. So at the late stages of the disease, the pathology is associated with a shrinking of the brain. And at the cellular level, you see these these aggregates, or these build-ups of clumps of protein that occur in between the cells. These are called amyloid plaques, and they're made up of aggregates of clumping together of amyloid protein. You also get clumping of protein within the nerve cells themselves, and these are made up of tau protein, and they form tau tangles. However, this is kind of what you'd expect to see at a late stage in the disease. I'm actually interested in what predisposes people to getting the disease in the first place. So there is a number of kind of risk factors that increase your likelihood of getting Alzheimer's. Age is the highest risk factor, but there are also things like lifestyle choices, such as diet and smoking and drinking, cardiovascular risk, diabetes. But what I'm particularly interested in is genetics. And so I'm going to kind of walk you through how people, this is what work other people do, other groups have done, looking at the genetics behind Alzheimer's disease. So to do this, what they do is they identify a large number of individuals who have the disease, who have Alzheimer's disease in this case. And they also identify a large number of individuals who are of a similar age, but who do not have dementia or do not have Alzheimer's disease. And what they do is they take DNA samples from each of these individuals, and then they sequence the DNA from these individuals. So they basically look at the code of the DNA. And then they compare the DNA from the people with dementia to that and they compare that to the people without dementia. And they're basically looking for differences in the DNA code. So the example shown here is in the controls, so that in the top DNA strand, you can see that in the controls there's a GGC versus in the dementia individuals there's a GAC. So you can see there's a single DNA change here in the dementia group. So okay, this is an example of a one base pair change, but these studies look at the entire DNA, so look across the entire genome. And the data is kind of summarized here. I'm not expecting you to fully understand everything on this graph, but every dot represented on the graph is an example of a single DNA change that's kind of present in the population. So we all have DNA changes in our DNA. That's kind of what makes us unique and different from each other. But the majority of those DNA changes don't actually do very much. So what they do is they compare the people with dementia and without, and they look to see if there are DNA changes that happen more often in people with dementia than the people who don't get it. And that's basically highlighted by this kind of blue area, these blue dots at the top. So these are, each dot here is a DNA change in the upper part of this graph, that's associated with an increased risk of getting Alzheimer's disease. So it's great. The geneticists have identified all these DNA changes that increase your risk of disease. But the question is, what do these DNA changes do? So as a quick revision or reminder, what does DNA do itself? So DNA codes for genes. So the DNA code, it's made up of genes, and the genes are transcribed into RNA. RNA is translated into protein, and protein really kind of dictates what the cell type, what cells are, so how they identify and how they function. So the human body is complex, as we know, is made up of multiple organs, and each organ has very specialized cell types. But the conundrum here is that each of these cells basically shares the same DNA. So how can you get all these diverse cell types when they all have the same DNA? So the human genome, or our DNA, codes for over 20,000 genes. However, not every gene is expressed or switched on in every cell type. And it's thought that different combinations of genes being switched on in particular cells is what dictates the identity of that cell and the function of that cell. So I kind of put like a little bit of an analogy here with the light bulbs. So here there's 5 different light bulbs. So imagine each light bulb represents a gene. And if you take a nerve cell here, for example, you can imagine that gene D is switched on, whereas the other genes are switched off. And this gene could be important for how a nerve cell functions. Whereas if we were to look at an immune cell, perhaps gene D would be switched on, and instead gene B would be switched on. Okay, so that's great. So another question is, does our DNA only code for genes? And you're probably wondering why I'm asking this, but you'll find out in a minute. And the answer is no. So the genes are kind of separated from each other. They're not like adjacent one after the other. They're kind of, there's space between them. And these kind of regions between the genes are called intergenic non-coding DNA. So they're basically DNA that does not code for proteins. They don't contain genes. So why are these intergenic non-coding DNA regions interesting? That's because most of the DNA changes that increase the risk for Alzheimer's disease that I mentioned earlier are not located in the genes themselves, they're actually located outside of genes. So they're located in these intergenic non-coding DNA regions. So I guess the question then is intergenic non-coding DNA just junk, which is what people thought it was originally? Or does it have a purpose? Does it actually do something? And the answer is yes, it does have a purpose. So within these non-coding intergenic regions are these epigenetic switches called enhancers. So in a situation like this, so here the DNA is represented by this black line, and the gene is shown here as this kind of blue bar, and then an enhancer that's in the non-coding region is shown here in green. And when the enhancer is not contacting the gene is switched off. So you can see that by this light bulb. However, you can get the formation of these DNA loops that bring these enhancers in direct contact with their target gene, and the gene gets switched on. So you can see the light bulbs on here. And our hypothesis or prediction, so what we predicted was that perhaps the DNA changes that are associated with an increased risk of Alzheimer's disease, if they're not in the genes, perhaps they're located instead within these enhancer regions. Perhaps they're actually located within the epigenetic switches that are needed to turn the genes on. And if this is the case, this could affect their ability to switch on the genes, and maybe you'll get genes that don't get switched on as brightly as they should. So the light bulb here looks a bit dimmer. Okay, so in summary, what do we know? We know that DNA changes that increase Alzheimer's disease risk are non-coding, so they're not located within the genes. We know enhancers are located also in these non-coding regions, also outside of genes. We know these epigenetic switches, or these enhancers, switch on genes that are important for cell identity and cell function. And enhancers are very cell type specific. That's how they go about doing this. So if you, the enhancers that you find in a nerve cell will be very different from the enhancers that you find in an immune cell and vice versa. So what do we want to know about these so what do we want to know about these DNA changes that are associated with an increased risk of Alzheimer's disease? What we really want to know about these DNA changes is which cell types are affected, and also which genes do they switch on? And to be able to answer these questions, what information were we missing? So what we didn't know were where are the enhancers located in different cell types of the brain. So for the different cell types that are in the brain, where are the enhancers located? And then also, what genes do these enhancers switch on? Okay. So before we can begin to do that, what we need to do is we need to get some brain tissue, and we need to separate the different cell types that we're interested in. And brain tissue doesn't look exactly like this cartoon. So as we know, brain is a complex organ. It's made up of multiple different cell types, including nerve cells called neurons, and also immune cells here called microglia. And brain tissue doesn't exactly look like this cartoon. This is actually what we receive from the brain bank. So this is a tiny little piece of brain tissue. And what we want to do is to be able to separate these different cell types, we need to mush it all up, which is shown here. And what we're actually interested in is, because it's very difficult to get intact cells out from frozen tissue, what we're really interested in is the nucleus of the cell. So these are the little circles that are in the middle of the cells here. And the nuclei or the nucleus is important to us because that's where the DNA is packaged. That's where the DNA is stored. So the first thing we do is we extract all the nuclei from all the cells inside the brain. And then we have a way of tagging these to be able to separate them or differentiate them according to the cell type that they came from. So we are able to tag nuclei that came from nerve cells versus nuclei that came from immune cells. And then we separate these according to the cell type of origin by using an approach or a technique called fluorescence activated nuclei sorting. So I'm not going to go into details about this, but this is basically what the equipment looks like. And we have one of these here at Imperial. So we're able to separate these different cells. So great, we've got our different cell types isolated. Now we want to be able to map the location of the enhancers for each of these cell types. So to be able to kind of understand this, we need to kind of understand how DNA is packaged into the nucleus. So in this picture, the DNA is represented by these this black strand. And to be able to package this kind of long strand of DNA into a tiny little nucleus, it gets wrapped around these proteins called histones. So histones are particularly useful because they can be tagged according to the function of the DNA in that region. So very kind of useful for us is the fact that histones are tagged with a marker called acetylation in regions where the enhancers are located. So this histone acetylation, or the molecular tags on the histones, is basically acting as an epigenetic tag for marking where these epigenetic switches are, where these active enhancers are. So how do we go about actually figuring out where they are? So we do a molecular process called chromatin immunoprecipitation, which first involves cutting up the DNA into small sections. And this is where the epigenetic tags become really useful. So they allow us to separate the enhancer regions from all of the rest of the DNA that we're not interested in. So we then take this enhancer DNA that's kind of been purified and separated from the rest of the DNA, and we sequence it so we can get the DNA code of the enhancers. And we do this for all of the different cell types that we're interested in the brain, including the nerve cells and the immune cells. Okay, so now we have our enhancer locations for 4 different cell types of the brain, including nerve cells and immune cells. We also know from studies done by other people, the DNA locations of these, the DNA changes that are associated with an increased risk of Alzheimer's disease. And all we wanted to do then was to overlay the two, to overlap them. So do these DNA changes that increase the risk of Alzheimer's disease, do they overlap with these enhancer regions? And the answer is yes. So we did this for using the enhancers for the 4 different cell types that I mentioned before, including immune cells and nerve cells. And we looked to see if DNA changes associated with Alzheimer's disease overlap with each of these 4 cell types. We also did this for 2 other brain conditions, including autism and schizophrenia. And so to be able to interpret this kind of plot on the left here, dark blue indicates that there's a high overlap of these DNA changes with the enhancers of that particular cell type, whereas a light blue indicates that there's kind of a low overlap of the DNA changes with the enhancers or the epigenetic switches. So as highlighted here in this red box, so this is for the DNA changes that are associated with an increased risk of Alzheimer's disease. You can see it's dark blue here for the immune cells, indicating that there's a high overlap of DNA changes associated with Alzheimer's disease in the enhancers or the epigenetic switches of immune cells, and not for the other cell types. This is in contrast for autism and schizophrenia, where you can see it's dark blue here for nerve cells, showing that there's a higher overlap for enhancers in nerve cells instead. Okay, so we've now gone from looking at the DNA changes that increase the risk of Alzheimer's disease, and by using information on epigenetic switches, on enhancers, we were able to identify that microglia or brain immune cells are important for the genetic risk of Alzheimer's. So what we wanted to do next was figure out what are the target genes. Because as we know, the enhancers and these kind of DNA changes are not located in the genes themselves. So this is not as trivial as you might imagine. So these enhancers do not always interact with their closest gene. So in the example shown here for gene A and B, this green enhancer is in fact interacting with a gene that's further away, so gene B. So now we do some different molecular approaches where, again, we need to cut up the DNA, and then we do some molecular biology followed by DNA sequencing that allows us to identify where these loops are this time. So this is an example of the kind of data that we get from this kind of experiment. So this is a bit of a complicated slide, but I'm going to go through each of the 3 gray tracks shown here one by one. So in the top gray track, so highlighted here in this green circle, each of these dots are the DNA changes that are associated with an increased risk of Alzheimer's. So this is where the genetic risk is located. If you look at the second track, this red signal here, this red peak, indicates an enhancer that's found in immune cells. And we know that this enhancer is also not found in other cell types. It's specific to immune cells. And if you look at the bottom track, you can see these are the DNA loops, and that this enhancer region is tied to or looping to these genes that are further away. And you can see actually it's looping to three different genes, which are highlighted here in yellow. And if you look at the very top, you can see that these are actually real genes, and this is actually real data. So we did this for all of the DNA risks, all of the DNA changes that are associated with an increased risk for Alzheimer's disease. And in microglia, we were able to find that 50 genes are tied to these DNA changes. So in the plot below, each circle here represents an individual gene. And these, as you can tell from this kind of, hope you can kind of gain from this plot, these Alzheimer's disease risk genes are highly connected. That's because they actually kind of fall within similar pathways and have similar functions. These Alzheimer's disease risk genes are important for immune function, which is what you would expect given the cell type, and also in amyloid beta processing. So if you remember from my first slide, Alzheimer's pathology is associated with these amyloid plaques, so this also makes sense. So in summary, brain immune cells called microglia are important for the genetic risk of Alzheimer's disease, and genes that regulate immune function and amyloid processing may be impacted by the genetic risk of Alzheimer's disease. So these are all the different people that I've worked with during my academic career. So science is not done in isolation; it's done with a group, with many individuals, and I've had a privilege of working with a lot of amazing people over the years. And I just also wanted to highlight that an important part of doing science is communicating science, including giving talks like today and giving posters. And you also get to visit a number of interesting places and meet interesting people along the way. So yeah. Sorry, it's a bit long.
Thank you so much, Alexi, for your talk. Really, I love the step-by-step that you gave, and thank you for taking us along the journey of what you do to get that data. And also, I love the light bulb analogy. I think that was a really good way of explaining those enhancers. So if you have any questions for Alexi, please leave them in the Q&A box, and I will ask them at the end of the session. And I can see some really nice comments coming in the chat as well. So without further ado, we're going to watch our first video. This is a video from the Imperial Care Research and Technology Centre. And as you saw yesterday, not all dementia research happens in a lab with cells. There are different ways of doing dementia research. And one of them we heard yesterday was sleep monitoring, for example, from Dr. Lucia Lee. So here's another video from a group at the Care Research and Technology Centre. Thanks, Maria.
Hello, everyone. My name is Maria. I am a PhD student at Imperial College and a robotics researcher at the UK DRI CRT.
Hi, my name is Ting. I'm a postdoctoral researcher at DRI. Today, we're happy to show you some of our robotics research for dementia care.
Some of you might be familiar with social robots, or perhaps some of you have interacted with conversational agents or smart speakers like Alexa before. Essentially, social robots are intelligent machines able to interact with us humans through speech, facial expressions, or gestures. These robots can play a very significant role in supporting the well-being and independence of people affected by dementia at home, and if designed appropriately, they can monitor health and provide personalized assistance, for example, by giving us reminders or activity suggestions based on what we need or like.
In our team, we work with many different types of robots to investigate what target users prefer and engage the most with. This may include voice agents like Alexa, virtual robots, or physically embodied robots. Overall, our research involves a lot of coding, data analysis from interactions with users, and also speaking to real users, getting their feedback to address their real needs. Here, we present Minder Diary, the Alexa skill we developed that conducts a daily well-being questionnaire for people living with dementia.
You told me you were a bit annoyed last time we chatted. Did you feel more agitated?
No.
Great. I remember you felt content last time we talked. Are you anxious or worried today?
Yes.
I'm sorry you are feeling this way. Can you tell me why you are anxious?
I have a doctor appointment today.
Thanks for letting me know. I hope you feel better soon.
Here is an example of a virtual robot called BrainBot, which works on a tablet. It shows facial expressions, understands our human language, and supports people in taking blood pressure readings.
Hi, I am Bot, your digital assistant. This is our first chat. What is your name?
You are speaking to Megan today.
Well done on taking your blood pressure, Megan. I have a few questions to ask you. Shall we start?
Sure, go ahead.
Great, Megan. Let me start by confirming your readings. You can check these by looking at the phone app. Could you please tell me the systolic blood pressure? This is the highest of the two numbers.
Thanks for watching. Goodbye.
So that was Maria and Ting's video from the Imperial Care Research and Technology Centre. So thank you so much for both of them for making that video. So as you can see, if you're interested in things like computer science and coding, there's also, a place for you in dementia research. So for the next talk, we're going to have Blanca come and speak to us about astrocytes, which you've already been introduced to through Alexi's talk. Again, if you have any questions, leave them in the Q&A. And thank you so much, Blanca, for coming to talk to us.
So, let me share my screen first. Okay. All right. So, my name is Blanca. I'm a group leader. Hold on. Do you see a shade on the of the screen?
Yeah,
That's why
there
I
is
had
some.
this. See, we said it would work, and now it's doing weird stuff. There was a way to remove this, no? Here. Okay, now, right?
They're disappearing.
Sophie, it's working now, right?
Yeah.
Okay, so today I'm going to tell you a bit about my journey to become a scientist and to now run a lab in which we study astrocytes. And after that, I will tell you about astrocytes. And as I told you in the title, these cells are very special because they are called astrocytes because they look like stars, as you can see here. So each one of these little blobs is an astrocyte in the brain. And I'll tell you more a bit later why we think they are very important for brain function. So, okay, it's not moving. Let's see. Okay, so where I come from? I'm from Spain, from a city called Sevilla, in the south of Spain. And two things that I may tell you about Sevilla is that it's a very beautiful city with very nice weather, except in the summer. Don't go in summer, it's really, really hot. And also it has very nice food. And I think it's so nice because throughout the history, it has had many exchange of cultures. So you can see that in the architecture and you can also feel it in the food. So what things have happened in Sevilla? First of all, I was born there. Second, I went to university there as well. And when I went to university, so I wanted to mention this because sometimes people think that scientists know they want to be a scientist since the very beginning, right? But I wanted to be many things. And I didn't know if I wanted to study biology or architecture or I wanted to be an artist. And probably by this drawing, which I made, you are thinking that it was a good decision not to become an artist. And instead, I became a biologist because I really like to know how things work. And I thought it was very cool to understand how life works. So I still didn't know I wanted to be a scientist. However, towards the end of my studies, I was like, maybe I'll do a PhD in neuroscience because I was really intrigued by the brain and also by the molecules that make the brain work. So I became a PhD student. And during this time, I fell in love with astrocytes. And this happened a bit randomly. I wasn't studying astrocytes during my PhD. I was studying mostly neurons. But there was a scientist that came to the university to give a talk about these cells, and then it was, love at first sight. So that made an impact on me. I continued my PhD studies, finished, and then I was like, okay, I like doing what I do. I'll continue the academic career and do a postdoctoral research. So I moved all the way from Sevilla to Chicago, and okay, this was a different place, right? And here I'm showing you this. This looks very dramatic. Chicago gets a lot of snow, but I loved it because where I'm from, actually, this is a thing, right? Since before I moved to Chicago, I had seen snow twice in my life. So you can imagine how impactful was this for me. I love it so much. Chicago is such a nice city. So I went there, and what happened there? Well, I was there for a year or so, a year and a half, let's say. And I went there to do a postdoctoral research on neurons and psychiatric disorders. Okay, and here you see in this picture some neurons that I imaged while I was there. Neurons are very beautiful, but I mean, you've seen the astrocyte. I just leave it there. So things that happened in Chicago as well which was a challenge for me, is my native language is not English, right? So I had to learn how to communicate effectively in English. And I had learned some English in Spain, but it wasn't enough to communicate properly. So this, I always think about this happened in 3 phases. The first one is I learned how the pronunciation of some words that I know from written language was actually being pronounced. Then the second one is I learned to be fast enough to make a comment in a conversation on time, not when the conversation has already finished, because at the beginning I will spend several minutes figuring out how to say what I wanted to say. And the third phase, the final one, was when I felt I really could, communicate, is I could make jokes in English. And not just repeating a joke that I learned by memory, like I could actually make my own joke. So this was an improvement, a challenge, but very stimulating as well, intellectually. And the third one is I made life-lasting friends from many countries. And this is the beauty of science. You are interacting with people from all over the world. You are exposed to many different cultures, and it's really, really enriching. But I was still in love with astrocytes, and I was studying neurons, and I wasn't feeling fulfilled. So I decided to make a turn and moved from Chicago to Los Angeles, seeking the nice weather again, and also seeking my first love, which were astrocytes, and work on them for several years. So the things that happened in LA were, at this point, I was able to say most of the things I needed to. Okay, that was a good start. Second is I spent over 5 years investigating the astrocytes in health and in disease. And you can see here one of them with the cell bodies and thousands of branches that are able to interact with many other components of the brain, as I will show you later. Another thing that happened is I got into Chinese tea because I had a very good Chinese friend, and every time she will go to China, will bring me some tea, and I'm still addicted to it. And one day, my postdoc advisor came and said, Blanca, there is a position to run your own lab in Edinburgh at the UK DRI. And then I was like, okay, LA to United Kingdom, closer to Spain, definitely, that's a plus, but it's rainy. So I want to try this. And I said, okay, I love astrocytes and how they hack the blood vessels. So if being in Edinburgh gives me the chance to do that, I will do it. So I flew from LA to Edinburgh to interview. And this was my interview panel. And I came to them and told them the same thing. I said, I love astrocytes and how they hack the blood vessels. I will investigate that. And I showed them this. Well, I drew this for them. And they gave me the position. So after all, my artistic skills were useful at the end. And this is when I got the confirmation that I got the position. This is when I landed from LA, in this case to Sevilla, coming back home. And I get emotional because it was a very happy moment. And this is when I started my lab in Edinburgh. So that journey, all of it to study these cells. And why am I so in love with this? Why are they so cool? So look at this. Here you have two astrocytes. One in, well, both of them are colored in pink. They have a cell body with branches that come out of them. And then these branches branch out into very, very small branches. I hope you can see them in my mouse because I'm pointing at them. And then you can also see the brain. This is a slice of a mouse brain in which we can stain the different cell types. So you can also see blood vessels here in green, and you can see the neurons in blue. So you can already see in these pictures that astrocytes are in very intimate contact with the brain blood vessels and at the same time with the neurons. The neurons are the ones that we think send the signals to the rest of the body or receive signals from the rest of the body to control behavior. But the blood vessels are very important because they bring all the nutrients and also some signals into the brain. So we think that these cells that have a very unique position in the brain are able to sense what is happening in the blood and what is happening in the brain, integrate that information, and then respond back to it. So I think this is a very interesting position to be in, and I do believe if we understand more about this, the function of these cells, we could apply to eventually cure dementia. So this is a very close-up picture of astrocytes. We now go out and look at a brain slice from farther away. You will see that astrocytes cover the entire brain. All of these green dots and green fuzz are the astrocytes that are tiling. We say they tile because they are close to each other in the brain. So I'll play it again. And these tubes that you see here are the branches of astrocytes that are covering the blood vessels. So as I said, they are in very close contact with the blood vessels in the brain. So astrocyte tiling, what is this? This is interesting because astrocytes have their own territory. So here you see one in green and one in red. So they don't invade each other's territory. However, at the tips of their territory, they interact with each other. And what is interesting about this is that they not only interact with each other, but they have little tunnels that connect their insides. So actually, let me explain this picture. In this picture, what you are seeing is astrocytes in purple. And then in green, you see a molecule that I put inside one astrocyte. And you can see how this molecule can spread out. The white and the green is the spread out of the molecule throughout the astrocyte network, right? So each one of these little balls is the cell body of an astrocyte. So what this means is that these cells have maintained their own territory, so they can control what happens there, but at the same time can perceive what is happening in the rest of the network. So it's very, very complex behavior that they may have. What else? Well, the cells are interacting with all the other cells in the brain. I told you already they interact with the blood vessels, and this is the part that in my lab we are most interested in. I'll tell you a bit more about it later. I also told you they interact with the neurons. So why do they interact with the neurons? We know that they can sense their neuronal activity and they can regulate it and help them perform their function in the right amount of intensity. We also know that during development, astrocytes are involved in controlling how many connections are formed between the neurons. So they are important to regulate that the brain doesn't, the neurons don't form too many or too little connections. And in addition, because the astrocytes are in contact with the blood vessels, they can take up nutrients from the blood. They can digest those nutrients and make them into a form that is more useful to the neuron. So they kind of like feed the neurons with something that is more useful for them than whatever comes directly in the blood. So you can tell they have very supportive roles, right? They are also in contact, of course, with other astrocytes, as I mentioned, They are in contact with the microglia that Alexi mentioned before, the immune cells of the brain. And actually, astrocytes are also important for the brain response to injury. And they are also well in contact with the other cell types. So again, very complex cells. And the thing is that even though neurons have been studied for 150 years, astrocytes were discovered at the same time, roughly, than the neurons. However, they haven't been studied by scientists until 30, 40 years ago. And the reason for this is because we didn't have the right scientific tools to study them properly. So the development of new technologies in science is key for science advancement. So actually, many people involved in science are not scientists like I am or like Alexi. They are engineers, for example, and they help us move forward the technology for us to be able to answer the scientific questions that we have. So what things we could do with this technology? Well, one thing, this technology, the one I'm showing you in this slide, is very old. And this is just electron microscopy, which is a very sensitive type of microscopy with which you can see very, very small things. So with this technology, we can see that when neurons connect with each other, as you see these green neurons, astrocytes are very close to these connections. I told you they can regulate neuronal activity. So what this morphology is telling us is that the astrocyte is close because it's receiving this information and responding to it. As I told you, the astrocytes also are in contact with blood vessels. So what you are seeing in this picture is a blood vessel that is cut transversely. So the cell that forms the tube is this yellow one. This is the white thing is where the blood will go through. We have other cells that are called pericytes, and then we have the branches of the astrocytes in green that basically completely wrap the whole vasculature in the brain. Okay. The function of these branches is still not very clear. There are some hints about what they do. For example, that they can regulate the blood flow, so how much blood gets into certain parts of the brain, or that they can maintain the function of the blood vessels in the brain in other ways, but we still don't know very much about it. Another thing that we know about astrocytes and that compares to contrasts with neurons is that we knew that neurons have electrical activity, right? And we can measure this. We can put an electrode on one neuron, and then we can measure the electrical activity, which is shown like these peaks in this graph. We do the same thing with an astrocyte, which is this little ball here. I'm not sure if you will be able to see. Then this is completely flat, right? So you say, oh, astrocytes are boring. Oh, this is what many neuroscientists said for decades, right? Astrocytes are boring. They just don't have electrical activity. However, with technology, we were later able to measure calcium signals in astrocytes. So look at this video. So here, there are two astrocytes. This is the cell body. These are the main branches. Other one starting. Let me play it again. So what you see is sparks. They are sparking all day, all night long. More during the day than during the night, actually. And we believe that this is the way they communicate, through these calcium sparks. And we know this because if we put the astrocytes, we add ATP, a molecule that can be released by neurons or by other cells, then we see that the calcium signals will rapidly go up, see? So, you know, calcium signals are a way of communication of astrocytes. However, unlike for neurons, we still don't know what their language is. What are they saying through these signals? So this is something that the field is still trying to understand. What are the astrocytes talking about? And well, what do we do in my lab? All these things are things that we know based on studies from many scientists. In my lab, before I tell you what we do, this is my lab, right? So this is where we do the work. Here you have different machines. There's several centrifuges, which you heard of yesterday. They help us spin. You have, this is a machine to cut brain slices, small microscope. This is a shaker. We in the lab, we have machines that just to shake stuff. And this is our Eva. This is not Eva. This is Eva. So our microscope has a name, and this is the microscope that we use to take the beautiful pictures that I've been showing you. And this is the team. This is how the scientists look like in the lab. And this is where we were working in. Steve just joined like about a year ago. So as I said, we are interested in how astrocytes interact with the blood vessels. And the reason for this is because the blood vessels, well, the reasons, several reasons, but one of the reasons is the blood vessels are very special in the brain. They have something called blood-brain barrier. And what is this? This is that all these components in the vessels, including the astrocytes, control everything that comes in and out in a change between the blood and the brain. This doesn't happen in other organs in the body, but the brain is so sophisticated that everything that is a change needs to be controlled. Is a obsessive, right? So when this obsession with controlling the change fails, then neurodegeneration happens. And we know this occurs very early on in neurodegenerative diseases like Alzheimer's disease, for example. But we don't know why this happens. And we would like to know if there are ways in which we can correct it so we can prevent neurodegeneration from happening. Because when the blood-brain barrier is not tight, then this is toxic for the brain. So we want to find out how astrocytes play a role in this. And for that, we use mouse models, we use human samples, we will use cell culture. We focus mainly, as you know, in the astrocytes, and particularly in the branches of the astrocyte that wrap the blood vessels and the vessels themselves. We use microscopy like optical imaging or electron microscopy. We can analyze the morphology of the cells properly. And we also use molecular biology. So we think this is very important because the proteins, particularly the proteins, because the proteins are the machines of the cells. So if we can know what proteins are in the interaction between the vessels and the astrocytes, we could also know how they change in disease, and then try to see if we could correct those changes to prevent the disease. So currently we focus on aging because it's the main risk factor for neurodegeneration. We also look at inflammation, and we also look at brain vascular disease or models of Alzheimer's disease. So this is what I wanted to share with you today. Thank you so much. These are the current members of the lab, another astrocyte with its vessel. And also I listed collaborators that we have in the UK and also in the US. As Alexi said, science is nothing without collaboration. So yeah, and without sharing our knowledge. So thank you so much for listening.
Thank you so much for talking to us. Thank you so much for such a inspiring and touching and funny, very funny talk. I really, really enjoyed it. Thank you. And you can see in the chat that many people did as well. Keep leaving your questions for Blanca down in the Q&A below, and Blanca will be back later to take some of your questions. We're going to quickly move over now to have a look inside some more labs. This one was made by myself and my friend Sophie in the UK DRI Cambridge Centre. So Amelia's just going to play that now. Thank you. So that was an insight into what we do in the Cambridge Centre. This was filmed during COVID, so things have changed a little bit now, but hope you enjoyed that video. Okay, so we're next going to move on to our flash talks. The first one is coming from Amber from the UCL Dementia Research Institute, who's going to tell us about what the amazing things that technicians do in the UK DRI. So thank you so much for joining us and enjoy Amber's talk. Thanks, Amber.
Yeah, okay, let's press play. So, can you see me? And
yeah, that's great.
Yeah, okay. So here we go. Okay, so hi everyone, I'm Amber. I'm a research technician at the Bartels Group at UCL, and I'm presenting today on technicians and how we keep science running smoothly. Okay, so I come from a non-science background. I was actually a marketing manager before this, so I worked on designing campaigns for brands. I decided to go back to university to study neuroscience at King, so I did a bachelor's there. During my time there, I did summer internships and a final year project in dementia, and I was lucky enough to have the opportunity to be a personal research assistant to a few postdoctoral researchers who went on maternity leave and got a grant to be able to come back and hire a personal research assistant to catch up on their experiments. And during this, I learned that I really enjoy the support role, and I was able to specialize in multiple research techniques. And due to this, I decided to pursue a career as a research technician. So what I work on is Parkinson's disease, which is a neurodegenerative disease that affects motor neurons and eventually spreads throughout the brain, causing dementia. I work on the protein alpha-synuclein, which is a pathological protein in Parkinson's disease, and it aggregates, causes cellular dysfunction, cytotoxicity, and neurodegeneration. So, and here you can see the protein aggregating in the brain. So this is my area of work in the lab. And what I actually do is what I'll be talking to you mostly about today. And I think this week is a very good representation of what technicians do and my role in general. So this week, I'm working on a collaboration with a project from an Alzheimer's group in our lab that we collaborate with. So I'm isolating amyloid beta from human brain samples. So I get human brain samples in, I dissect them, I purify the protein out of the brain, and I inject it into this machine where I do liquid chromatography to separate out the amyloid beta. So I spent a couple of days of my week doing that this week. And then I'm also working on developing clinical tests for Parkinson's disease. So this involves collecting blood from patients, which I luckily don't have to do myself. We have lovely collaborators over at the Queen Square Brain Bank and UCLH who do that for us. And then I get the samples, I fix them, and I use fluorescent probes and different techniques to look at alpha-synuclein in these patients' blood. And hopefully, ideally, we will be able to use this sort of technology to have tests to see if people have Parkinson's disease pretty early on. And that's half of my job in the lab, and the other half is actually in the office. So what I'm doing this week is I'm preparing shipments for collaborators. So this involves me preparing international customs documents. So I need to know a lot about regulations from different countries on importing products and exporting human samples to them. Which has been quite an interesting part of my job post-Brexit, because there's been a lot to learn about new import regulations. I'm also setting up interviews. So we had a candidate for a postdoctoral position that we have this week, and they came in and I had to arrange catering, the room bookings, and make sure that everybody turned up on time and for their one-to-one interview with that person. I'm also preparing some monthly spending reports. So I do this every week because I'm the person in charge of the ordering and managing the equipment in our lab. So every month I tell my boss how much we're spending and more importantly, how we can save, because that is an essential part of what I try to do to keep my lab on the budget. And then the most enjoyable part of my job is mentorship. We have visiting students right now. One is from Germany. And I'm taking under my wing and teaching him about the lab. And that's what technicians do a lot of, is we're the person who you can come to. We know what's happening in the lab. We can answer most of your questions. So beyond our actual expertise is we sort of make sure that there's a person of contact that everybody can get in touch with and keep things running smoothly. So just about briefly what other technologists' roles. So you can become really hyper specialized these days in tech as a technician. There's a lot of advanced microscopy, gene editing. So as a career, it's quite attractive because you can spend an entire lifetime getting into these sort of technologies and becoming very, very specialized in them. And then also we need people in research management. So we have lab managers, senior staff scientists, and science administrators who keep everything going. So we need to have people in these roles as well in science. And then finally, a research technician role can be a very good stepping stone into a PhD. So if you're unsure about what you want to do, so you've heard a lot of talks here, and I'm sure a lot of it has been interesting to you, and you're wondering, oh my God, there is so much to do in dementia research. But if you're unsure of exactly what you want to get into, you go to university, you see some things that you're interested in, and then you say, I want to do a PhD, but what I want to do it in. And if you're unsure about that, that doesn't mean that you can't pursue a PhD. So if you took a technician role, you'd be able to get a lot of good skills in and then sort of figure that out as well. And a lot of technicians do end up doing that, is going into a PhD after. And you can save up money. And if it is if you're a really, really brave person, you can also do a PhD while doing a technician role, which is a lot to manage, but financially it can really work out. So yeah, that was my little 5-minute flash talk. And let me know if you have any questions in the Q&A after.
Thank you so much, Amber. That was fantastic and so good to see your perspective as a technician. And perhaps we have some future technicians in the audience here today.
I hope so.
Yeah. So yeah, keep leaving your questions in the Q&A. Amber will be back later. We're going to hand over to Tatjana now, who's from the King's College UK Dementia Research Institute. So thank you for coming to talk to us today.
Okay. Hello, everyone. Just bear with me 1 second while I share my screen. So I'm Tatjana, and I'll be talking a little bit about basically researching diseases of the brain while you're wrestling kind of with your own brain. So this is essentially a perspective of going into studying in academia with being neurodivergent and not really being aware of it, but more on that later. First, a little bit about where I'm based. So like everyone else, I'm also part of the UK Dementia Research Institute, and I'm all the way down here in London at King's College London. And yeah, this is our building. And basically, I kind of have a few different things that I'm doing. So my training, as you will see later, is kind of as a biochemist. What I'm actually doing right now is RNA biologist, but the title of my PhD is kind of basic in clinical neuroscience. So it's like a big mix of different things. And next to my PhD, I'm also kind of quite active in the PhD community, representing students within my department, but also within my faculty. And that's something that I'm very passionate about. So in terms of what I'm actually doing in science, you're probably familiar with this, which is the central dogma of biology, where DNA gets transcribed into RNA and then translated into proteins. So I'm working with both proteins and RNA. So I'm specifically looking at RNA binding proteins and how they interact with RNA in health and disease. So you might remember from a few years ago, the Ice Bucket Challenge. Here we're seeing Benedict Cumberbatch undergoing this. And I'm showing this because the diseases that I'm focusing on is amyotrophic lateral sclerosis, ALS, and frontotemporal dementia. So, and I'm focusing on a set of 3 proteins. So that's FUS, the pink one, and then TAF15 and EWSR1. So in a healthy person, these proteins are normally localized in the nucleus of the cell, and they're imported from the cytoplasm with this receptor here. But then in ALS, FUS is mutated and it aggregates in the cytoplasm, as you can see here. And in frontotemporal dementia, it's all 3 proteins that aggregate in the cytoplasm, and kind of the underlying mechanisms of why this is happening and why these diseases then play out the way they do. It's not quite clear, but there's some indication that it's probably an issue of RNA metabolism, because all these 3 proteins, they're RNA binding proteins. I'm not going to go into too much scientific detail of kind of the methods, what I'm doing, but sorry. Essentially, my work is mainly based in cell culture. So you've seen people probably do some mice work, or what we've seen in some of the earlier presentations is like slides of brain or something. So I don't really do anything of that. And this is why sometimes I'm a bit, I'm a neuroscientist, but I'm not really a neuroscientist. But yeah, I work with cells. So these are HeLa cells and SHS5 cells, which we also like to call sushi cells. And 2 of the main things that I've been doing during my PhD, you can see one up here, which is called RNA fish. So basically you're using molecules that emit light to find your protein in this case, or RNA in this case. And the RNA fish, so that's specifically targeted just for the RNA. So these are fluorescent molecules that bind to my RNA. And then for the protein, I just use like an antibody to show me kind of the localization and where it is. And in this specific experiment, I was trying to see on the top here, you have your normal protein as it's expressed. So you saw in the previous slide, like normally it's nuclear, and that's what you can see here. So blue is the nucleus of the cell. And then my protein and my RNA both kind of co-localize with the blue area. So they're all in the nucleus. And then when I express my mutant protein, you can see that the protein and the RNA, they're now no longer in the nucleus. And so that's kind of what happens in disease. And what I was trying to figure out or show with this experiment is that it's not just the protein that mislocalizes, but also the RNA. And then another big part of my project involves just looking at expression of RNA in general. And I'm also looking at part of the RNA metabolism called splicing. I'm not sure if that's something you've already encountered. And a technique that I use to do that is reverse transcription qPCR. So basically I take RNA and then I reverse transcribe it to cDNA, which makes it more stable. And then I use this fancy robot here to kind of mix different reagents. I then put samples into this, and I end up with plots kind of like this that show me different kind of expression levels of different RNAs. And yeah, so you heard the word RNA a lot of times in this, and that's why I would call myself more of an RNA biologist instead of a neuroscientist. But yeah, that's kind of the things that I do in my day-to-day work. Yeah. And now a little bit about my journey and my background. So I'm originally from Switzerland, and I did my bachelor's and master's in Bern, Switzerland. And during my bachelor's and my master's, I had projects in both, and I kind of switched topic a number of times. So in my bachelor's, I was doing some research with Trypanosoma brucei, and then in my master's, I was working on CRISPR-Cas9, but also doing kind of RNA biology. One thing that happened to me, which is now where kind of the ADHD being neurodivergent part comes in a bit more, is that I've always been a really good student in like elementary school and secondary school, and I just absorbed information like a sponge, and I wanted to learn about everything, and I was just so passionate about like any kind of new things. But then as I got to university, basically what happened is that my grades that were usually quite high and like on a good level, they suddenly dropped, and because I just could not focus on all the subjects anymore. Like our education was kind of like so broad, and it was so much information that just from sitting in the lectures, that was like very difficult for me to absorb the information, and I did not know what was going on essentially until kind of like last year, which is something I'll get into in like a second. So as my grades kind of went down, this was kind of a little bit my motivation for university. So I had this big dream of like going to all these amazing colleges, and like going to the #1 college for like neuroscience and all this kind of stuff. And I started kind of giving up on that a little bit, because I felt like I would never have the grades now that I would need for that. But then as I went on, I kind of found some new motivation eventually, because I wasn't ready to kind of let go of this dream. And with a lot of hard work, I then ended up here at King's College London. And basically, yeah, so it required a lot of hard work. And I did not have a student life, as a lot of people kind of, maybe if you think of university, you think about like students going out and having a lot of fun. I did not do that. I stayed at home and I studied a lot. And then what also helped me to get essentially my PhD position is like through connections. So if you have the chance, if you do decide to study, do not hesitate to reach out to people and ask them for help or ask them for guidance. It's something that, so I ended up having 2 offers, like one in London and one in Munich, and both of them I had from just like reaching out to people and being interested in their science and talking to them about their science. And yeah, so that's something that I would give on, and have a lot of patience. And in terms of like, in generally handling potential suspicion of maybe like not being able to learn or process things as other students would, if you have the suspicion, do try and reach out to someone. Like I wish kind of that I would have done this sooner, because it helped me a lot within the past year to then be able to work with people from the university. Because for example, King's College, they have a great learning and disability service. So they were able to help me put measures into place that allow me to do things a bit more my way. Because when you have something like ADHD, it's not that you're kind of like damaged or non-functional, it's just your brain functions into a different way. So yeah, it was super helpful for me to then eventually reach out and talk about it and get to this. And maybe as like a quick summary, this is like loads of things. So yeah, I switched research topic and locations throughout my academic career, which is something that I found like highly beneficial, and I would recommend this to you as well. And it's also great to, if you're thinking of maybe eventually starting a PhD, to get like some lab experience, either as a research assistant or in an MSc. And then when it comes to actually studying in your undergrad, don't feel forced to, do what everyone else does. Find something that really works for you. If it works better for you to maybe study from home and to read the books or learn the concepts as it is, that's something that you could try instead of forcing yourself to go to lectures where you're not able to focus or not able to kind of absorb any of the information. And then network, and if you can, like, try and do some extracurricular activities, because they can also help you network and find a different kind of passion, and then help you motivate yourself more again for studying. And finally, if you do struggle with poor focus and memory, you're not lazy. It might be that, yeah, you have some specific learning difficulty or ADHD, and so do try and reach out to people that could potentially help you get that assessed. And then here I just have like a few memes, and I don't know if one of you struggles with this. Maybe you feel, I don't know, spoken to by any of them. I love this Instagram account. And yeah, that's it from my side. Thank you. If you have any questions after this, also do not hesitate to contact me either on Twitter or by email or anything. And yeah, thank you for listening.
Thanks, Tatjana. That was such a great talk, and thank you so much for sharing your journey. And just like Tatjana reached out to some other people, feel free to reach out to her, and you've been invited to. So thank you. And we're now just going to pass over to Christina, who's going to share her last flash talk for the day. So
yes, I'll share my screen. All right. So I'm Christina. I'm a PhD student in the Home Lab at the DRI. And well, today I hope to teach you a little more about microglia, and also, yeah, tell you about my experience as a researcher. I'm from the Netherlands originally, so that's the neighbors across the channel. And well, when I was around your age, I guess, I was doing something very different. I was mainly doing this. I was a member of the national synchronized swimming team in the Netherlands. And I was traveling a lot, participating in tournaments. And well, in the end, we didn't make it to the Olympics, which was a shame, but that sort of made me focus more on my studies. And there was a lot that I was interested in, and I couldn't really choose what to do. And basically, yeah, the only thing that I knew that I did not want was to become a scientist and to be in biomedical research. Well, that turned out very differently. And that was mainly, I think in hindsight, I just didn't really know what biomedical research was and what it all entailed. And it's, I guess, great that you guys are tuning in here now, because yeah, you are learning the things that I didn't know at that point. So back to my title, Microglia as Scientist. So in order to explain that, I think we should first look into what a scientist is. And what I do on a daily basis is, yeah, reading stuff about my topic, microglia, learning from the research of others. And well, I also prepare samples in the lab, and like you've heard, I guess, from many people now. And that's never a lonely task. There's always your lab mates around you. And well, when you've prepared your samples, you take these in my case, to the microscope, and I look at the microglia that you see here in green. And all these images you can run through software, and then you can analyze whatever you want to basically see. So you can answer the questions that you have with the data that you collected. And in the end, well, hopefully you can also share your results, whether positive or negative, with your lab mates, or at conferences, sharing it with your international colleagues. And well, basically all of this, what a scientist does, is also what a microglia does. So you've already heard about microglia. They're the immune cells of the brain. And well, they really do the same, because they also explore their environments. They have like specific receptors to catch up on cues like proteins or lipids. And sometimes whatever they take up, they can also process. And it's kind of like preparing a sample. And they analyze that. And they also work together with a lot of the cells that are around them. For instance, the astrocytes that wrap the blood vessels, other immune cells, and they really try to keep your brain like a healthy place. And well, data collection and analysis is maybe a bit far-fetched for microglia, but the samples that they process in like inside their cell bodies, they can trigger like biochemical reactions and cascades of reactions, and that in the end makes them decide to, for instance, share their results. So they can show that they found a certain protein which may be a threat, and they can signal to other cells that something is going on. So microglia and Alzheimer's disease, we touched on this already too, but yeah, we don't really know what is going on with these microglia and Alzheimer's disease. There are signs that they're doing good stuff. So they're clearing the protein aggregates that seem to accumulate over time in Alzheimer's disease patients, their brains. And on the other hand, we also see that they can damage neurons. So they can eat the synapses, which are the contact points of the neurons. So they are very crucial for the neurons to do what they're supposed to do, which is signaling. And well, you know what mRNA is by now. So in my project, I measure mRNA levels in the brain, and we have fancy techniques right now to actually measure these mRNA levels in an intact brain, so without tearing apart the tissue. And that gives a lot of information about, well, the mRNAs, the amounts of mRNAs in specific cells. And we think that based on that, we can deduce what those cells, and in our case, the microglia, what they are doing. And hopefully we can then find out which microglia are the good ones and which ones are the bad ones in Alzheimer's disease. That was it. Thank you. Yeah, just let me know if you have questions or anything. Happy to answer them.
Thank you, Christina. That was a fantastic talk. And I love how you are a microglia and so fantastic. So before we move on to the Q&A, I'm just gonna share my screen. So as you heard, some fantastic talks today. You might be aware that the recordings will be available both from today and from yesterday's session as well. Before I head to the Q&A, I'm just going to do this last little bit before, in case people need to rush off. So in your inbox tomorrow, you will find hopefully some links to the recordings, as well as links to resources about dementia research and careers and research itself. So some resources we've collated for you. In addition, you will find a link to a post-event questionnaire, and that will take you just 1 minute, but it will really help us design next year's event. And so if you could fill it out, that would be fantastic. Before I move to the Q&A, I just want to say a massive thanks to the UK DRI communications team, Molly, who's been doing a lot of the promotion, Amelia, who's right there running the show behind the scenes. It doesn't just happen by itself, these kind of events, and there's loads of people involved as well, who aren't necessarily doing lab work or anything like this, but who are also involved in making dementia research run. A team of volunteers from across the Dementia Research Institute, so this is staff and students, and these are the people who help promote the event and send out lots of emails so that you managed to find this event today. And thank you also to the Follow Me Around video makers who made these videos. So if you could please fill out that post-event questionnaire, that would be fantastic. So I'm just going to quickly now jump to our Q&A. So if all the speakers would like to come back and turn their videos and mics on, that would be brilliant. Yeah, so maybe Blanca, I saw that there's one for you straight away. So why does adding ATP increase the calcium signals, which were so beautiful, those videos?
So, okay, why is because there is a receptor on the surface of the astrocyte that when it binds to ATP, it sends a signal to the inside of the cell and tells the components inside the astrocyte to release calcium, because the astrocyte has some reservoirs of calcium inside it. So when the ATP binds to the receptor tells the reservoirs to release this calcium. So this, we know how it happens. What we don't know is what that is what that triggers, right? So we know the calcium increases, but we don't know what happens after that and why is it important for the astrocyte to increase the calcium inside of it. So more to be understood.
Thank you, Blanca. One for maybe Alexi and also Christina. Is there a happy medium with microglia where they don't do damage but still help? I think it's a really good question.
Well, I guess, yeah, that's a hard question. I mean, everyone's after that, I think. And in the end, I guess it's multiple microglia. And yeah, I'm not sure whether you would agree with that, but I guess it's multiple types of microglia that all work together. And yeah, it's like when you're in a team, like everyone does a different task. And yeah, I don't know, some balance others out. And I think it will probably be something like that.
Yeah, no, I agree. I think so. I think the issue is you want the microglia to react somewhat to changes in the microenvironment. So if there's like cells dying and debris and these protein deposits that need to be cleared, the microglia need to react to that. And then the problem is, the way I understand it, although I don't really look at microglia behavior at the cellular level because I'm always looking at DNA, is that the issue is that they then become kind of hyperactive or too activated, and they start chewing up things that they shouldn't be chewing up, like synapses and things like that. So yeah, it's kind of trying to get them to react appropriately, but not overreact.
Yeah, no, I would totally agree with that. Yeah.
So Hannah, who asked that question, you have a very good question that potentially you could be answering in the future. Maybe one for Amber. Would you clarify, is a research technician role similar to a lab manager role as well?
So there can be, depending on the size of your lab. So as a research technician in a smaller lab, you will take on a lot more of managerial roles. So similar to what I do, things like helping with budgets, doing shipping. So these kind of things that lab managers traditionally would do. So there's a little bit of a acceleration that you can do basically. So go from a research technician who's hyper specialized in certain techniques, or a research technician who manages the lab as well as does experiments, which is sort of where I'm at, doing half and half essentially. So yeah, they are similar, but they can be distinct as well.
Thanks, Amber. Okay, so we got one yesterday, which I think I would like to ask to you as well, is what you would say to your 18-year-old self. And I think that's a really nice one to hear. So if anyone wants to start off.
Okay, I can start. I will say, what I would say is just follow what inspires you. Do whatever you feel inspired by. No matter how difficult you think it is, if you like it, then I think you'll manage.
I would say to myself that life is very long. Coming back to science after having a separate career, when I was 18, I thought, oh, you have to pick exactly what you want to do and do exactly that. And this is the path. And if I don't pick now, God forbid, what would happen? And that was exactly how I was, very intense, and turned out completely differently. And you can have many different careers, and life is very, very, very long. And it will be longer the more that we work on dementia, because we're going to extend life even more. So you don't have to make the right decision at 18. There's still lots of time to get it right, and then get it wrong and get it right again.
Yeah, I guess I would like to tap into both of yours, your comments, and maybe add to that, you know, there's never a wrong choice. Like whatever you choose to do, you always take something away from it. And I guess in the end, I made a lot of detours, like different projects, different fields, different things that I was doing in life. And yeah, I don't know, in the end, I'm really happy that I did all of that. And I don't think that I ever made like a wrong life choice or something. I think you always learn, and I think that's the most important. And it probably brings you even further if you learn from many different things.
Yeah, I guess I haven't spoken. So I agree. I think with what Blanca said at the very beginning is just do what you feel like is what you really want to do and in your heart, and don't worry too much about like external things. So I guess for me, I was like, when I was 18, I was very confused about my sexuality. I was a little bit worried about what people in different sectors would think about that. It was also a different time. I went through the education system where teachers were not allowed to weren't allowed to talk about LGBTQ issues. So I know things have changed, but then I also realized that there's other challenges that I don't understand that you guys are going through, and to just not let those become a block, or try and do what you actually really want to do in your heart. That's what I would say.
I think I would totally agree with that. And also, like, it's gonna sound very cheesy, but like, don't be afraid to dream big. And if someone else tells you that you can achieve this, like, don't believe them. Just keep working, keep doing your own path, and don't let other people distract you. Like, you can reach whatever you'd like, as long as you, stick to it and work hard for it. And sometimes there's also just like a little bit of luck that's needed, like you just know the right person or something like this. But definitely don't let other people push you off your way. Like, stay focused and you'll get there.
Thank you. I feel like that's a really nice place to end on. And if our speakers are happy to share, like, would you be happy to take any more questions from the audience in your Okay, brilliant. So thank you so much, everyone, for coming again. And thank you to all of our speakers for their fascinating and inspiring talks. I think we can all agree. So without further ado, we're going to call Making It Brain 2022 to a close. Thank you, everyone, for coming. It's so nice to see so many people from everywhere. And look out for that email in your inbox and for the recordings, higher quality videos and the resources and that questionnaire. And thank you so much again, and have a fantastic weekend. Bye.
Thank you.
Bye.
Thank you. Bye.